All right, good afternoon, everybody. Happy Thursday. Hope everybody's well. Hope everybody had a good lunch. Ready for that long weekend? We're going to finish up nucleic acids today. I'll give you a few minutes if you didn't finish lipids, because I'm about to put the scores for the lipids in the grade book. So if you didn't finish the Nearpod lipids, make sure you do that. So I'll give you a few minutes for that while these other things come up. We're going to, so bring up your macromolecule chart because we're going to look at, we might get through both the nucleic acids and water and be done with the chart today. That's the objective, is to finish up that chart. All right. So again, take out your macromolecule chart. Let's see. And we're looking, and again, go to Canvas, and we're looking at getting nucleic acids and water done today. I am adding the scores for lipids in today, so if you haven't done that, make sure that you finish doing that as well. We were doing lipids yesterday. If you were absent, and I don't think anybody was, the code for the Nearpod is in the assignment folder. What is that? Oh, I don't want that. All right, I was trying to get Go Guardian, but instead of Go Guardian, it gave me something else. Okay. All right, I think everybody's here today. Good, good, good. Nucleotides, nucleic acids, and water done, and we'll be done with the chart. All right, she's in. Yay. Good afternoon. Good afternoon. Macromolecule charts, that's where we are. One more, 'cause okay. Ready to go. All right, remember you're supposed to be on camera. I got 3 of you who are not on camera. Everybody should have their macromolecule chart up. You should be in Canvas. That's where we're going. I think Charlie is not here. And yeah, the internet has been doing some funny things. Um, this morning, every time I tried to share, the Zoom kicked me out. So be aware that if it kicks me out, I'm coming right back. So don't leave. But yes, the internet has been an interesting thing today. Okay. All right. Getting there. We're getting there. Still missing 2 people, but we're getting there. Okay. All right. So I am going to share my chart. And again, we're going to be flipping back and forth between Canvas and the chart. And again, we're looking at this from the standpoint of it's a macromolecule. Later on, we have when we do protein synthesis, we're going to have a whole lesson on how proteins are made. But today, we're just interested in the fact that nucleic acids are one of our 5 very important macromolecules. Okay, so again, the reason we're looking at them is because living things can't do without them. They're not living as a unit by themselves. They're just elements connected together. And my chart doesn't seem to be up. That's why you have it. So yes, I'm aware the screen is clear because it's clear on my side too. It's trying to load my macromolecule chart. Let me see if I can switch pages and move it from here. Yeah, there's a oops thing going on here. All right, I'm going to refresh this page. Let's see if we can do that. Yeah, the internet has been interesting today. I don't know. You're missing Nearpod? What Nearpod are we missing? Yes, go ahead, go to the restroom. Dallas, I don't know what you mean by the missing Nearpod. I mean, I need to go in and get your carbohydrate scores. Is that what you're saying? I need to go in and check on upgraded scores. I will do that. I will do that. I don't know what's happening with my macromolecule chart. It won't come up. All right, I'm going to switch out and do the module. Hopefully you got your chart up. Okay, I'm going to go in and check. Yes, Dallas, I'll go in and check those and upgrade because a couple of y'all are missing. I will update. All right, so everybody, wow, the share screen is very slow. All right, so everybody should be here. This first one is just kind of a introductory thing. It doesn't even talk, it just has slides. We have looked at, so this is what I need you to have on your chart. And I really wish I could pull mine up because I have this on my chart. In the first column under function and purpose, you need to have this. All right. So you can copy and paste that and put that on your chart. So everybody should be in Canvas so that you can follow along with me. In your first column under purpose, what is the function of nucleic acids? Now, what I kind of need to backtrack a little bit. Up here, it says DNA and RNA are 2 examples. You can put that in your last column. Okay, examples, where you have examples, put DNA and RNA. So there's 2 types of nucleic acids, DNA and RNA. Then you can come back to the first column and put down that they're function and purpose is to carry out the genetic materials. Let me try one chart. There we go. Open the chart. Like you said, the internet today is doing whatever it wants to do. Now it doesn't want to open it. Open. All right, well, while mine is thinking about opening, I'm gonna come back over here to what you need to have on your chart. All right. Under your second column, so we already have pictures of the nucleotides and the polymers. So we'll get as soon as my picture comes up, I'll get back to those. In the column, in your column that has special things, you want to write these two things down here. Okay, this is what goes in the next column. All right, so there are 4 possible nitrogen bases. Try it one more time to get this thing to do. Yay! Finally. All right, so now I can show you. All right. Like I said, every time I share, I lose my Zoom. But I'm back. So we will get back to looking at, now that I got the chart up, now I got to get the Zoom to stay put. And actually, let me stop sharing because I forgot to click the sound. Share sound. Click on that. Okay. Hopefully it'll stay put for longer than a minute. Okay, so in your second box, like I showed you, there should be 4 possible nitrogen bases. DNA has adenine, thymine, cytosine, and guanine. And RNA has adenine, cytosine, guanine, but it has uracil instead of thymine. Okay, so each of them has a nitrogen base. Now, if you need the picture for your chart, let me know and I'll put it in the chat. In fact, let me just go ahead and put it in the chat. Oops, that's not what I want. Okay. All right, that picture. So you need that picture on the nucleotides. All right, so remember monomers are the single unit. So we're going to take a bunch of these single units, which are made up of phosphorus, a phosphate unit, a sugar, and a nitrogen base all connected together. All right, so you see this thing over here where it says nitrogen base? In the other column, we just said there's 4 different kinds of those. So it's always the same phosphorus. The sugar depends on whether it's DNA and RNA, but those are what we call the backbone. And then we change up the nitrogen base. It has to be one of the 4 different kinds. Okay, so a nucleotide is composed of a sugar, a phosphate, and a base. Not all 4, just one. But you know this, that there's 4 different nucleotides because there are 4 different bases. All right, then we put the nucleotides together and we make our polymer. See, I'll put that in the chat in case you don't have that. And that didn't work. So let me try it again. Copy. There it is. All right, so the picture's in the chat for those of you who need to have the picture. All right, and in the last column, you should have that there are 2 examples. RNA and DNA and RNA are 2 examples of nucleic acids. Okay, so that's what you need for your chart. So everybody's chart should have that much on it. So what we're looking at here is with RNA, it's single-stranded, so it's just one row of nucleotides connected going down. With DNA, it's double-stranded. And like I said, we'll talk more about this later. Right now, you just need to know the difference between DNA and RNA is one of the bases is different, and one is single-stranded and the other is double-stranded. Okay. All right, so we're going to, everybody good? Anybody not good? All right, now again, remember, this is a study sheet for you, so don't just complete it and then don't study it, because this is what you need to know about new macromolecules. Also, you're going to make sure that you save it, because at the end of the year, when you have to take your Georgia milestones, you're going to need this information for that as well. So don't make it disappear. Put it in if you have to create a subfolder from when you change from one semester to the next, make sure that you put it in a safe place. All right. So let's go back to Canvas. All right, and we're gonna watch this little video, and then I'm gonna give you time after the video to do the practice box, and then I'm gonna do attendance while you're doing the practice box, and then we're gonna do water.
Hi, this is Ms. Delosier, and these are your notes on nucleic acids, so DNA or RNA. So nucleic acids, DNA and RNA, right? We're all familiar with those. That is your hereditary material. It's your hereditary information. It's on your chromosomes. It's in your genes, whatever you want to say about it. It's your nucleic acids, all right? And they're made of monomers of nucleotides, all right? So DNA is made up of 4 nucleotides, adenine, thymine, guanine, and cytosine. And then RNA is made up of A, G, C, and U. And we're going to review that again in a second. So let's look at what a nucleotide is first. So a nucleotide is made up of a sugar to start. So we have a sugar, and in DNA, our sugar is deoxyribose, which is a 5-carbon sugar. In RNA, our sugar is ribose. So let's look at this 5-carbon sugar, and then we're going to go ahead and we're going to attach it to a phosphate group. Now, because that phosphate is negatively charged, this is going to provide an overall negative charge to my DNA, which means DNA is most definitely charged, which means it is most definitely going to be hydrophilic. So that's my nitrogen base. Okay, and so that all 3 of those together make up a nucleotide. When we did these in class, I just had you make the base because we didn't have large enough kits to make the sugar and the phosphate group too. A yellow sugar, or you're going to have a yellow phosphate group attached to a white sugar, right? So yellow, white, yellow, white, yellow, white. So basically what I have is I have this backbone going down the sides that's phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. And then hanging off the sides, you end up getting the nitrogen bases. And the nitrogen bases, because DNA is 2-stranded, right? The nitrogen bases stick off towards each other and they form like the rungs of a ladder, and they bind together through hydrogen bonding. All of the bonds in what I've got drawn here are covalent bonds, but when I actually have like A and T next to each other, they're going to form hydrogen bonds. Okay, they're not going to actually form a covalent bond across the strands. They actually form many hydrogen bonds. So that's my DNA structure. That's a single-stranded structure, but we made it DNA. It's a single-stranded DNA. So let's talk about the 5 types of nucleotides. So my 5 nitrogen bases, there's only 5 nitrogen bases. There's adenine, which always pairs to thymine in DNA, and then guanine, which always pairs to cytosine in DNA. And then in RNA, I have uracil, okay? So adenine and guanine are what we call purines, which basically just means that they're double ring structures. So see the double ring structures below? Like they don't.
Okay, I'm gonna pause it here for just a minute. What she's telling you now, you don't have to know for this unit, but we will need to know this later when we do DNA and protein synthesis. You will have to know who matches with who and who's replaces who and what the 2 different kinds of molecules are. But for now, just take that as information and store it in the back of your mind. But this part right here, you don't need to know for this unit.
Memorize them. That's a waste of your energy. But they're double ring structures. Adenine
Like
and guanine are double rings. Thymine and cytosine and uracil are what we call pyrimidines. And I always remember this because uracil is the shortest of the nucleotides, and it's a single ring. So single ring, but it's the long word. Pyrimidines sounds like it should be that double ring thing, and it's not. So I just remember that I'm wrong all the time, and that the shortest word, uracil, is a single ring and goes with like the long pyramid type thing. That's how I remember it. I don't know how you're going to remember it, but you need to know that. So uracil is going to bind to adenine. So if I have an adenine in my DNA, when I make an RNA copy of that, it would go A to U. And we're going to do activities on that in class. So those are my 5 nitrogen bases. And so in DNA, I'm going to have a double strand, and I'm going to have A, T, G, and C. So I'm going to put like a little yellow check mark next to all four of those. And then in RNA is single stranded, and I'm going to have A, U, G, and C. So I'm going to put a pink check mark next to A, U, G, and C. And so that's your basics on the monomers of nucleic acids. Um, and that's really all I want to talk about on DNA and RNA for these notes, because we're going to spend like an entire unit on DNA and RNA, and like how we
All right,
actually
so I'm
make.
going to close because that's right. We are spending a whole unit on DNA and RNA and all that stuff that she just said. But basically you need to know as far as your chart is concerned. Let me pull my chart back up for a sec. All right, so you see how we have just the one little nucleotide? There are actually 4 different nucleotides, and they're named by whatever the nitrogen base is. Okay, so there's 4 different ones for DNA. There's 4 different ones for RNA. So while we have a picture of just one, there's 4 different kinds. All right. That being said, that's all we need on our chart for DNA and for nucleic acids. So take a minute to go through this. I'm going to do attendance. I'm going to come right back. This is quiz-worthy material, so make sure that you get that right. I'm going to go over it with you as soon as I stop doing attendance. So do that, and then if you finish that, jump ahead to water, because that is our next and last macromolecule. Okay, so you work on that, I'll work on attendance, and I'll be right back. All right, and I promise I am going to go, as soon as class is over, I'm going to go update all your carbohydrates, lipids, and protein Nearpod scores. So make sure that that is something, you have something there that I need to see if you don't already have it done. Progress reports go out next week, so we want to make sure that all grades are up to date. All right, Charlie and Desiana. Mm. All right, everybody should be working on the practice. I don't think this person's in. Okay. 3:12. Okay. Okay. Right now, nothing makes folks come to class faster than marking them absent. Okay. All right, so hopefully you did and I'm going to go back to sharing. Hopefully my screen won't disappear. All right, so if you did the matching, the click and drag, again, these are quiz-worthy questions, so make sure that you have actually done it. I'm going to show you the correct answers in a minute in case you didn't do it, but you should have checked the box to make sure that you got the correct answer. So these are the answers that you should have. And of course, if you checked it, it will, oops, so no, it just downloads an answer key. Oh, there you go. All right, but it will download a actual physical answer key when you click answer key. All right, so that's what you need to know. It should be on your chart. And we're going to move on to water. So water again is our last macromolecule. Now water is going to be a little different. All the ones that we've looked at so far have carbon, hydrogen, and oxygen in them in some kind of way. Oh, we didn't add that to our chart, but let's go back and do that. Because that's important to know. And I think I explained to you that the terms organic and inorganic. Organic means it contains carbon. So let's go back here. We'll make it a little smaller, just a little
Miss,
bit. All right. So since yes,
I can't find the water lesson.
question. Okay, it might be that I need to publish it. Give me 1 second. But before we do that, so like how we have on the chart that they're made up of carbon, hydrogen, oxygen, carbon, hydrogen, oxygen, carbon, hydrogen, oxygen, and some phosphorus. Put down carbon, hydrogen, and oxygen and nitrogen on this. So this one is like, yes, I'll give me 1 minute to go to the Canvas page and tell you what you should have there. But yeah, go ahead down here and add carbon, hydrogen, oxygen, nitrogen, and phosphorus. All right, so those are the elements that are, now when we get to water, all we're going to have is hydrogen and oxygen. Okay, whoops, there we go. That's all water is made up of. So notice what it's not made up of. There's no carbon. So everything else that's important to living things has carbon in it. And that's what makes them an organic material. Water is extremely important to us, but it's an inorganic material because it does not contain carbon. Okay. While I'm here, let me put these molecules in the chat, and then I will figure out what's wrong with Canvas for you guys. Okay. It might be that it is not published. Oops. Oh, shucks. Undo. Yay. All right. Copy. There we go. All right, so that's our monomer and our polymer. All right, I always say the one water molecule looks like Mickey Mouse. And then we have a bunch of Mickey Mouses hooked up together to make our polymer of water. Okay, so I put those in the chat. Let me check Canvas for you to make sure that Canvas is actually loaded. I mean, published. And it is not, but it is now. All right, so what I need you to do, most likely, is to go out and come back in. And then you should be able to find water. Okay, so go out, come back in. Oops, come on. And water should be there. Okay. All right, this is a lab that I used to do just at the beginning of the year. If you haven't done it, it's an interesting activity to do. And I used to do it just as an activity on following directions and just on using the scientific method to explore how many drops of water you can put on the face of a penny. And the way it works is you take an eyedropper and you count how many drops of water you can get on the face of the pen. I used to have it as a challenge to see who could get the most drops of water on the face of a penny. And it's because water has a particularly funny characteristic to it, and you'll talk about that when you talk about water, I guess in physical science or whatever. But it's called surface tension. So where the molecules want to hold together until they can't hold together anymore. So you can get a lot. Some people get like 70 drops of water on the surface. Some people, it depends on how you drop the water. And so you can get 25 drops, you can get 50 drops. I've had somebody just squeeze real tiny drops and get a lot of drops, but that you will eventually get to the point where the surface tension bursts and all the water will spill. So the lab that I have is always called the drowning penny, because the penny will drown when surface tension breaks. Okay, so that's what that little thing up there is representing. And if you ever did the lab, you really would be surprised how many drops of water that you can get on the face of a penny. All right, so we're going to look at the characters. So you can put this in your first column. All living things depend on water. So let me go ahead and do that too. I keep saying I'm not going to do it, but I might as well. All right, so put that in your box, first box. All right, all living things depend on water. That needs to go there. All right. You don't necessarily, well, you do need to know, but not necessarily for biology, that it is 70% of the earth. It is the most abundant compound in living things. So that part we need to know. All right, so your body, other than being made of proteins, lipids, carbohydrates, is mostly water. So let's put that statement on our chart. Where did my eye go? Okay. Okay. All right. Will we ever run out of water? We will not, because water is part of the water cycle. See, I told you, just mark someone absent and they'll show up for class. There we go. All right. So again, this might have been something that you studied when you did physical science or life science, but there's a water cycle. And most of you know the three parts of the water cycle: evaporation, condensation, and precipitation. So we don't run out of water because it keeps cycling and cycling and cycling. We have low periods of water at times. A lot of the water, most of the water on our planet is stored up in the icebergs, and they're melting because of global warming, but we're still not going to run out of water. It's just that what is accessible to us sometimes becomes very low. What are the ways we can save water? Well, that's in the certain activities that you do. Um, don't run the water while you're washing the dishes, brushing your teeth, take shorter showers, that kind of, all those wonderful things that they tell you that you can do. And so this is something that we are going to actually look at later on when we look at cells. We're going to talk about when you put a cell in salt water. Ocean water is salt water. So we're going to look at what happens when you put a cell in salt water. It actually shrinks up. So you become dehydrated because all the water that's in the cell is out of the cell, and that's not a good thing. So no, you don't want to drink that. All right, so let's look over here. So we have our molecules. Um, 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 um. Okay, we have this thing called a pH scale. Okay, on the one end of the scale, so it goes from 0 to 14, and in the middle is neutral. Okay, on the low end of the scale, everything is acidic, and then they've listed the things that are acid. And on the high end of the scale, things that are basic. And again, when you get to chemistry, you actually to know this and know why something is an acid and why something is a base. We don't need to know that. You just need to know that water needs to be right there in the middle. You don't want water to be an acidic and you don't want it to be a base. Okay, so water is kind of in between 7 and 8. So they have seawater as an 8. Water needs to be neutral. Okay. You don't want your water to be acidic or basic. Okay. So read that. What else did we skip? Don't worry about this so much. Okay. It does explain acids and bases, and we're not going to worry about acids and bases right now. Again, just know that on this, there is a scale that we call the pH scale. These are the things in comparison as far as acid and base is concerned. And water should be in the middle. All right. Water, most of you know water is H2O. And so if you don't know anything else, you know H2O. And that means it's made up of 2 hydrogens and 1 oxygen. So sometimes we refer to water as HOH. because we have to take into consideration what the molecules actually are. So there's 2 hydrogens and 1 oxygen, looking like Mickey Mouse. All right, so practice this for a minute. I said water is composed of, how do we do this? Is it going to move? Oh, you have to submit it. Okay. All right, there we go. All right, next question. Water is unique because it is a polar molecule. Polar molecule means that it has a positive end and like a magnet. Magnet has 2 poles, a north and a south. Water molecules have a negative end and a positive end. So it's called a polar molecule. So put that, let's put that in on our chart under, since we have nothing over here. All right, so let's put that over here. Water is a polar molecule. Okay. All right, so if I make this picture just a little bit bigger, you can see that the hydrogens are positive and the water is negative. So this is what we mean by polar. Like I said, think of a magnet, think of north and south pole. There's a negative end of the water molecule and a positive end of the water molecule. The oxygen is the negative end and the hydrogen is the positive end. Okay. All right. Okay. All right. What property of water explains why it floats? All right, has to do with density. These seem to be all right, we can put down that put what it says in this box. Solid water, meaning ice, is less dense than liquid water. So put that, copy and paste that if you can, or just type that in the same column. Solid water is less dense than liquid water. All right, let's put that on our chart. Do we need to put in brackets ice so that you know what we mean by solid water? Okay. And you know we're not going to have examples of water because water is water. So there's no examples for that. Unless you want to, or we can do, okay, yes, we can. Examples, so we'll put ice and we'll put, I'll just say water or liquid water. Because water exists in all 3 states. Everything exists in all 3 states, but we want to say ice is solids. Then we have liquid water. And then we have a gas. Did I spell that? Sure did. Then we have water vapor. All right, all right, that's the best we can do for examples. That it exists in 3 different states. You can't say that with these up here. So what makes it different is you can have solid water, liquid water, and gaseous water. Okay? All right, so let's finish our little matching game over here. All right, we submitted. Okay. The property of water that explains large bodies of water maintain their temperature. All right. Okay. Oops. I guess that was the last question. All right. Water has a very high specific heat. So let's add that to our chart. Um, oh, I changed questions. Okay. All right. Just put down that it has a high specific heat. So let's put that on the chart. And we'll explain what that means later. And then, all right, so that's the first one. So this is a different question. So what property of water explains why liquid water does not evaporate until it reaches a high temperature? And it's high heat of vaporization. Okay. Okay, so it was this B answer. It has high heat of vaporization, which means it takes a long time to vaporize. And then we'll answer the next question. So let's put that on the chart. Okay. All right, if I move too fast, that's because it's the B box, high heat of vaporization. All right, next question. The property of water that explains why water cannot dissolve non-polar substances such as oil and fats. All right, and it's going to be that last box. I'm not going to click on it yet because I want you to get it. Water is hydrophobic. Okay, so that means it repels a lot of other molecules. So you know that um, if you put oil in water, it sits on the top. It does not mix. You can stir it all day long and all the oils come right back up to the top. Hydrophobic. Phobic means it doesn't like things. So it does not, it's not attracted to these other molecules. All right. Water is hydrophobic. So we're going to put that on the chart. Okay. All right, which feature explains why the attraction of like molecules such as water are attracted to water? Um, adhesion. Oops, didn't like that one, huh? Cohesion. Picked the wrong Hesian. Whoops. All right. This one was cohesion. I mean, the other one was cohesion, and this one is adhesion. Yeah. So the first one was cohesion, and this one is adhesion. They changed the question really quick. All right. Which feature explains when water rises? Because, all right, this is what happens in plants. So we're not going to write all these things down on our chart, but you need to. Capillary action is when, like when you suck on a straw. So the straw would be like a capillary, and the water going up the straw is capillary action. Okay. Explains why water is a good solvent because of its polarity. I guess. Universal solvent means that most things will dissolve in water. Most things dissolve in water. That's why it's called, yeah, I got that one wrong and I didn't like that. All right, so practice that one. Don't be like me. So go through and practice those again. It was that one that I got wrong with cohesion and adhesion. So make sure you do those correctly. But we're not going to add those to our chart, but make sure you know all of these properties of water. All righty. So read this down here. Again, I'm not worried so much right now about pH scale. Just know that water should be neutral. That's what you need to know about water. It should be neutral. And then know all the other properties that we put on the chart. So again, we have finished our chart. So make sure that you study the chart, know the chart, because guess what? Next week, we are finished with this module. That means test. And so I see you on Tuesday, because Monday is a holiday. So the Tuesday schedule is Monday schedule. So I will see you on Tuesday. All right. At the end, let me publish this too. All right, publish. All right, you can do a study guide. So take a look. If you get bored over the weekend, look at the study guide, because this is what we're going to look at next week. We're going to look at this study guide. And we're going to look at our macromolecule chart. And by next Thursday, we'll be ready to test. Okay? All right, so this is our objective for Tuesday is study guide and review for test. Okay? So we got like 3 minutes. You got any questions? Anybody need me to repeat something? Show the chart again? All right, so I did publish the part of the module that takes you to the study guide. They have it in Word and what is this? There's two versions of it, Word and PDF. Okay. So you should be able to open up one of those, and we are going to go through that as a study guide in preparation for the test. So we have completed this module. Can I post my chart? I'm going to try. Last time I tried to do that, it didn't work, but I'm going to try. So I'm going to save it as a PDF and see if I can't upload it now that we've finished it. Okay. All right. That's what I'm going to do, folks. I'm going to do my best to see. I tried saving it before and it did not exactly cooperate with me. All right. Let's see. Copy, share. Um, I'm gonna figure out a way to save it as a PDF so that I can upload it. Because right now, this program is not letting me do that. But that's my homework. Okay. So you can look for it. Mm, all right. I tell you what, let me try that and see if you get it. So I copy. I know. There we go. Export. Download as a PDF. All right, let's see. Let's see if that worked. So I'm going to put that. Where did it do it? Oh, there we go. Okay, so now, all right, so I have created a module just for, if I can get it all the way back up to where we are. All right, it should be right underneath yours. And for mine, I have to go through all of this, but up, there we go. No, where'd it go? Place else. Let me say move module. Move module. After. Ah, it's always an easier way to do things. Okay, there it is. All right, so you should have this now. You have a copy of my macromolecule chart. Okay, there you go. All right, any other question? Any other question? Because we are over time. So you don't need a link, you just need to go into, does anybody, so I published it, so it should be showing now. So in the module, it should come underneath the other module for macromolecules. Okay, you should have. All right, so I'm going to rename this. I'm going to do. Okay. But anyway, it's there. Okay. We good? All right. You have a great weekend. Bye. Enjoy your break. Oh, there it is. Edit. Mm, okay.